So I guess we should start, huh? So I'm Bob Murphy. Welcome to day 3. And I'm going to introduce for you the topic of Cell Organizer, which is our system for learning and models of the organization of cells, that is, the organization of the components of cells inside the cell. And when we talk about doing spatially accurate cell simulations, which is what our 3 days are about here, the question is, where do we get accurate spatial distributions, or spatial, distributions, in order to do spatially realistic simulations, right? Now you've spent 2 days learning how to do those, but where do we get those simulations from? And what I'm going to talk to you about, or where do you get those geometries from? And I'm going to talk to you about that. And related to that is the question of how do we explore the effects of any condition, mutation, drug, treatment, on cell organization, how that perturbation happens, and of course we also may want to incorporate that into any simulations that we do. So you've talked about spatially accurate modeling systems, but most of the geometries that are used in those systems, and by the way, I should have said at the beginning, please interrupt me if you have any questions anywhere along the line, come from either painstakingly hand-segmented electron microscope images, or from essentially cartoons drawn by hand, or with circles inside of circles or something, and as a result, those two sources are either inaccurate or very hard to generate. Right? So what we're going to talk about is Cell Organizer, which can generate geometries for doing cell simulations computationally. All right, so the basic idea, we know cell organization. This is what we actually see in a light microscope when we label a particular protein that's, for example, in the Golgi complex, or on lysosomes, or in this case, in microtubules. But the idea then is, if this is the input that we get from fluorescence microscopy, we would like to be able somehow to relate that back to whatever it is, whatever the cell structure is. And typically, the traditional way in which we learn about where proteins, organelles are within the cell, particularly where proteins are within the cell, is we tag that protein with a fluorescent dye and then take some images. And then an image like that gets converted into words. So you can imagine the complexity of this in terms of how many bits of information are there. But the net result is, in the vast, vast majority of cases, we're going to then take this and convert it into a small number of words like, that's a lysosomal protein. In fact, it'll be the Go term, lysosomes. That would be it. And that has traditionally been done by looking at the image and saying, oh, that looks like a nuclear pattern or a Golgi pattern or whatever. And of course, it assumes that we know what those patterns are, that we already have words for them in our mind. And it assumes that people are good at doing that task. Now, it turns out that last thing is not true. People are not very good at doing the task of figuring out what the organelles are that proteins are in. Now, the alternative is to use machine learning methods, and we can design numerical features that describe the pattern, like the lysosome pattern we saw, and then, again, assuming we know what the patterns are, use examples of those known patterns and teach a machine to do that task. And the machine turns out to do it quite a bit better than people do. And then, if we do that for lots of proteins, we have information that would allow us to put those proteins into a cell model that we want to do a simulation of. So you want to do a simulation of the biochemistry of some pathway, and you need to know where all the pieces are. You could then look up in a database, you could do that now, and you would find these Go terms that would tell you where each protein is, and you could make some kind of circle in a circle, sphere in a sphere model, or draw whatever you happen to feel like, or painstakingly segment an EM image, and then you could do some kind of simulation based on that. But here's the problem. This approach is descriptive, and it's extremely reductive. We take the complex pattern of a protein in a cell and reduce it to a couple of words. And then how do we, if we're trying to do simulations, how do we map it back into what it actually looks like in a cell? Okay? Because we can map it back into these cartoons, but we know that's not the way it actually looks in a cell, so what's the point of doing spatially realistic simulations if they're not spatially realistic? Or, again, this very limited approach where we can try to hand segment high-resolution images. Now, so there's an alternative. This is basically taking the problem of figuring out where every component is in the cell, where every component is in the cell, and using a descriptive approach for that, those words. Okay? And that's fine if all we're trying to do, which is the way most people initially envision this problem, is this kind of mapping into an idealized space of lysosomes, Golgi, and so on. And like if we wanted to classify apples and oranges, all we need to do is measure their color, and we're done, if that's the only two things we had. But if we're trying to make an apple, which is what we're doing when we're talking about cell simulations, we're trying to reconstruct and be able to understand the properties of it, we need what we would call a generative model, and that's what I'm going to talk about. So what do I mean by a generative model? Think back to when you learned your numbers and your letters. All right? You were given examples of a 5. You had two choices. You could have memorized every 5 that you ever saw, and if somebody asked you, write a 5, you would pull one of those 5s out of your memory and copy it directly down. Right? But that would not work very well if somebody asked you to recognize a 5, but you could fix that because you could try to estimate the distance between all the 5s you've memorized and each of the 5s you're trying to recognize. All right? But it turns out that's not how you do it. What you learn is a mental model of what it means to be a 5, which includes 5s that are not very typical, that are just kind of on the edges. You might have seen somebody, a handwritten 5, they're just barely a 5, but your mental model includes that with low probability. So that if you are to draw a 5, somebody says draw a 5, you pull that 5 out of your mental model. You can think of that as statistical sampling from the model of 5s. And then you draw one. So there's a really low chance you're going to draw like a really crooked 5. Most of the 5s are going to be pretty reproducible, pretty recognizable. Right? So you have a generative model in your head of what it means to be a 5. Now the task we're going to try to do is to learn to make a generative model of what a cell looks like. Now this is not learning the biochemistry, this is not learning the reactions that are going on. This is just what's the geometry, what's the organization of a cell. That's the task. And just like we learned this from images of 5s, we're going to learn that model from images of cells. All right? So the idea is to try to build the equivalent such that that generative model could be sampled from in order to synthesize cells. And now you can see where we're going, because that's exactly what you need if you're going to try to do cell simulations. Okay? All right, so again, what do I mean by this? What do I want to learn? This generative model of cells. Think of it as I have a test tube full of cells that maybe I just trypsinized off of a dish. And I'm going to spill those out onto a tissue culture dish. And they're each going to adopt their own morphology. They're going to have their own cell shape. They're going to have their own distribution of organelles and so on. Well, we can think of the model from which they are choosing. All right? They're doing a whole bunch of dice rolling saying, should I be exactly like this? Should I be like this? You know, what should my geometry, where should my organelles be? They're sampling from the generative model of cells of that particular type. Let's assume they're all the same cell type, fibroblasts or something, right? That's what we want to learn, okay? And then when one of them, you know, settles in, it picks an instance from that generative model. And now it's, and again, ignore time for the time being, right? So imagine that that got locked into that morphology. That's an instance. And we could take individual images of that cell and take many of them, for example. We might take 3D images. We might take images at different angles. There's all kinds of things we could do. But from all those images, we could do a very good job, a very classically well-established thing, to reconstruct that instance. So we could get a high resolution, for example, we're doing storm or some, you know, some high resolution microscopy. We could get a very good capturing of that particular instance, high resolution image, right? That is not the same as from many cells learning the model that gave rise to all of them, okay? That's what we're interested in. We're not trying to reconstruct individual cells. We're trying to build the model that generates all the cells in the first place, all right? And obviously, that is a ridiculously complicated task, okay? And I'm not trying to tell you today how to do that task to, you know, in all of its nuances. But I am going to talk to you about some ways in which we can start to use and that are useful as they are right now. So again, just to emphasize, that is to learn the underlying reality that, and not, by the way, that's an important thing. When we take images like this, they are not the reality. That's one of the problems with using images as basis for simulation of cells. An image of a cell is not really the reality. There's noise in there. There's a particular, you know, focal plane that you're looking at that cell through. The cell is actually much, for example, it may be hard to see where the membrane regularities are in the surface or on the cell because of noise. So that's just a view of that cell. So the first thing we want to do is learn the underlying reality behind that. That's what that reconstruction does. But then, again, the goal is to go beyond reconstruction to the generative model. We want to remove the noise but keep variation. That's a very important thing. This model captures variation of cells, right? It is not just one cell. Okay, so what's the general principle that we're going to try to use? The principle is going to be that we're going to take lots of cell images of some particular organelle, protein, whatever it might be. And I'm going to talk to you about this, but what we're going to do is we're going to try to find generative parameters that would allow us to reconstruct each of the cells from those parameters. So let's imagine that we're going to, and that's going to involve a lot of assumptions. So let's imagine, for example, that we're going to say cells are spheres, period. The only parameter they have is their radius, okay? So from each cell, we're going to estimate its radius, and that is a generative parameter, because if we know the radius, we can generate the sphere, right? That's not speaking to the question of whether or not a cell is a sphere. It's on the basis of assuming that it's a sphere, we can estimate the parameter of the sphere, the radius, okay? And then, so we do that, and that's what we're going to talk about specific examples of how that's done. But the basic idea then is, just like, for example, if we wanted to, reconstruct the population in the room, we could measure height and weight and arm length and legs and everything, right? Those are parameters that we could construct at least stick figure versions of all of us from, right? So do you, yeah, go ahead.
Just to understand, the model that is built based on statistics of many images, or this is an assumption and I just try to then measure it?
So there are two pieces. One is how good is a particular model for representing its instances. Like the sphere, not a particularly good model of cells, but so that's one question. How well does the model fit the instances? Yeah? The second issue then is how well can we build a statistical model from the parameters of the instances?
So the model is not built statistically from many, many images?
It is, in fact. It is, absolutely. It is going to, but so it's going to be two stages, right, where we're going to take each instance, learn its parameters, and then build a statistical model of all of those, okay? And that's what this says, right? So we're going to take those, so that's going to give us a row for every cell image, a column for each of those parameters. We're going to tabulate that, and then we're going to build some kind of statistical model, unspecified as of yet. But here's the key. Once we have that statistical model, we can sample from it, get instance parameters, and then synthesize images. Or we can stop calling them images. We can call them distributions or geometries, because they are idealized geometries for the cell. You, Tom.
Yeah, but to follow along with what Jan was saying, could there be a way to, and point you made, could there be a way to take the instances that are generated, generate synthetic images of those?
Yes.
I guess maybe the instances are images.
Yep.
And then do some unbiased comparison of those instances to the images.
Oh, to the original instance images?
Yeah.
Yeah. Okay.
In a way that is free of parameters.
Okay. Well, we're going to talk about parameters or not parameters, and like what kinds of models we can build. And there actually are non-parametric models we can use too.
Yeah.
This describes parametric models,
Yeah,
but
where you're not imposing any model.
That's absolutely correct. We're going to talk about non-parametric models also, okay? And I'll explain how that works. But what I want to get clear here is that there is the error that is introduced when I choose a model to represent my instances. That's the reconstruction error, right? That's the difference between the actual instance I had and the representation of it that I made.
So here,
Yes.
Here, you say sample new parameter. What kind of parameter is
Those are going to depend on the split. We're going to talk about that, okay? This is just in broad strokes, right? We're going to get to what those might be. An example was the radius of a sphere, right? Or the height and arm length and leg length of a person or something, right? And yes, question.
So you talk about randomly sampling new parameters and taking into account possible correlations between.
Absolutely, right? So you have reconstruction error of the instances, and then you have error in the statistical model to capture the instances, which might, for example, under or overweight correlations, you might, right? Whatever that statistical distribution is, might be a good one or a bad one. Right? So those are two distinct sources of error or variation. Yeah? Well, it's really error if you want to think of that, not variation. Okay, so that's what cell organizers are about, is to do this task. And here's the fundamental organizing principle. Obviously, images come in on the left. The training process is going to be broken down. Right now, we don't have a methodology whereby we could take a single model type and fit all of the components of the cell all at once. Okay? You can imagine that would be crazy hard. But what we do is try to simplify the task to some degree, maybe the way that a cell does, which is we're going to build a nucleus first. We're going to build a model of the shape of the nucleus. And then we're going to build models of the cell shape, and then we're going to build models of organelle shape, and then we're going to try to put them all together. All right? So what we're going to have is modules that depend upon each other. So the nuclear shape model doesn't depend on anything right now, the way it's set up. But the cell shape model is going to depend on the nuclear shape model. We're going to talk about that. So the basic idea to get out of this is that what we're going to have is modules that are going to learn particular components of the cell, and then we're going to mix those components together. Okay? And of course, then we can synthesize things. All right, parametric models, we start with nuclear shape. Now this, I don't want to go through all of the pieces that are in Cell Organizer, but I want to, yes.
Sorry to interrupt you, but
No, please.
a lot of the examples I've seen of this show these kind of flat cells that look like they're coming from culture. And I was just wondering about the practicality of going from kind of a more two-dimensional cultured cell to a three-dimensional, more structured, maybe like a neuron or something. Is that even feasible? Or
Yes, absolutely. In fact, we have models of neurons. I mean, when we talk about this, we typically show two-dimensional examples, but the vast majority of the models we build are three-dimensional, even for cultured cells, because even cultured cells have 3 dimensions. But there's absolutely no reason why these, and we use them, for example, for plant protoplasts, which are spherical and don't attach at all, right? And we use them for lymphocytes, as we'll see, right? So there's nothing in these concepts that are restricted to any kind of particular.
Sure, I didn't. Conceptually, it makes sense that you should be able to do any number of.
Right. Now, what's going to happen is that the choices that you make of the model components might differ.
And the number of components.
Exactly.
Huge, I would
Yep.
imagine, with departmental.
Exactly. That's right. Right. So what we're going to do is an illustration of how we could make a parametric model of something like nuclear shape. So here is a three-dimensional image of a nucleus, of a real nucleus. And we're going to do a simple coordinate transform from this Cartesian coordinates into cylindrical coordinates, right? So that we have the z-slice here, and then the angle here, and then the radius, or the distance from the center to the edge of the nucleus in each slice at each angle is here. So that the cell is, the nucleus is wide here, then narrow, then wide, and then so you get big, low, big, right? And that's just a coordinate transform, totally reversible, right? If I have this, I generate that. So now we're going to make our first parametric assumption, because this and this both have the same number of points, roughly, which is lots, right? Thousands or tens of thousands of points in that nucleus. We're going to say those little bumps on there are not important. Now, we have no real reason whether to know that or not know that, but we're just going to say the bumps aren't important. And therefore, that we can fit this with a spline surface, which has 32 coefficients, and that's a reasonable representation of that. So that introduces our first thing, our reconstruction error, right? That this is not exactly this. But the point is that with these 32 coefficients and a height, we can generate nuclei. Was there a question back there? No. Okay. All right. And so we're going to take those 33 parameters and learn some statistical distribution. It turns out that a multivariate Gaussian does a pretty good job of capturing this. You can test that. We'll talk about that later. And then we sample from that. We can synthesize new nuclei, right? That are representing the nucleus we learned. Okay? Yeah? So there is our nuclear model. Now there are lots of different ways we can learn a nuclear model. That's just one of them. So then we can learn a cell shape model. Now, we could have done that the same way. We could just model a cell individually, the cell shape individually. But then if we sampled from a nuclear model, an instance, and we sampled from a cell shape instance, we wouldn't know where the nucleus goes inside of the cell. We wouldn't know where to put it. Furthermore, we might have picked a big fat nucleus and a small skinny cell from these independent models, and they wouldn't actually fit together at all. So we have to have the models not be independent but dependent, and therefore we're going to learn a dependent model. And one of the ways, so that's what we're going to do with these arrows, is we're going to say the cell shape depends on the nuclear shape. And one way, a simple way to do that is just to make a ratiometric model where we take the distance from the center of the nucleus to the edge, the distance to the edge of the cell, the ratio of those two things, do that over 360 degrees, and learn that as a statistical model. Okay? So now that gives us, once we've picked the nucleus, then we can pick a cell shape to go with it. All right. Then now the fun part comes in. So that's what we call a framework. That's the basic model. You have a nucleus and you have a cell membrane. Now we're going to talk about models of organelles. Now, here's where we're going to also introduce the idea that is one of the real motivating factors behind Cell Organizer, which is that when we take an image of a cell, that is not the actual distribution of the organelles or the proteins within that cell. There's noise, there's sampling involved in that image. All right? And so, for example, if we label a lysosomal protein and look at high resolution, with a confocal microscope, not with a storm or a super resolution microscope, this is what we get. This is a picture. Now, this is a picture of a portion of a cell that has that's labeled for lysosomes. We could say, let's just learn that as, for example, a texture model. If you look at it, you kind of, what's a good model to represent that, right? Probably, some kind of texture where you're sampling individual pixels, maybe. But that's where we add another parametric assumption. We know lysosomes are membrane-bound organelles. Right? They're continuous membrane-bound organelles. So we're going to assume that this is actually a bunch of membrane-bound, sort of spherical tubular lysosomes, and we're going to fit this with Gaussians, multidimensional Gaussians, to say the Gaussians represent the real reality, the underlying reality below the image, and the image is just a manifestation or a view of that reality. Now, we're going to have to do that by the standard approach, which is try to make like the maximum likelihood estimation. Right? And that's what we do. Yeah? So once we've done that, that is a parametric model of lysosomes, and we can remember things like the number of lysosomes, the size of them in X, Y, and Z, how fluorescent they are, and so on. Right? So that's a model that will allow us to generate individual lysosomes. Now, here's an important point. They are, at least with this model, those are geometrically defined. So we know where the boundaries of those things are. We know the continuity. Right? So that if we want to do a cell simulation with something like MCell, we can say, I know exactly where this thing begins and ends, and I don't have to try to guess like I would here. If I were trying to do a compartmental simulation based on an image like this, it would just be, some sort of leaky blob, and there wouldn't be boundaries on this thing. Does it make sense? That's a very, very important point, right? Okay. All right, so we then have that. So we have the distributions of the objects. The only thing we need now is to put them in their places in the cell. We know how to sample individual organelles, individual lysosomes, mitochondria, peroxisomes, endosomes, whatever it is. We have these vesicular organelles. We need to know where to put them. So we're going to define a geometry-independent coordinate system, which is measure the distance from each object to the nucleus and to the cell boundary, and then that's going to be a map of where all the organelles are. We do that for all of the organelles in all of the training cells. So we take like 100 cells. That's going to be thousands and thousands of lysosomes. And then we build a probability density function of where a particular lysosomes are likely to be. Okay? This is showing that relative to the major axis of the cell, which is how we're going to define the coordinate system. Okay? And again, that's specific to a particular cell type. Right? And then we get a 2D density function. Right? And then we can synthesize, organelles, mitochondria in this case, from where we synthesize the mitochondria individually, and then we place them. And in this case, we're assuming they're independent. Now, we're going to talk about that in a minute. but we're going to assume we just can place them wherever we want. There's no relationship between their positions. But we could, in fact, learn that relationship. Now to non-parametric, right? So those were both, sort of good examples of very specific assumptions we're making about the nature of the model. And we can, yes, question.
Why do the reconstructed cells look like the V-stack optical slice is still visible?
It's a great question. It's because that's what we learned it from, right? But we can take that out. So that's a very, very good question. When we learn the model of the cell shape, for example, in those models, we're learning those ratio models in each slice. So we're building up the cell in slices like pancakes, right? We can interpolate that, and if you ask Cell Organizer for a higher resolution version, it will interpolate it and give you a smooth Z.
So like for the top slice of the V-stack, where you know that you're not capturing the curvature of the top of the organelle, does that reconstruction with higher resolution also try to capture the curvature of the top?
Yeah, if you had more slices so that you were capturing that, right, you could get that.
What if you can't, many microscopes you can't get more resolution?
That's correct. And so that's where it's going to interpolate, right? It's going to try to find a smooth, whatever interpolation function you pick, it will try to smooth those boundaries out, right? And it turns out that is important for doing simulations, because otherwise you get this sort of stair-step effect, and you get corners. Yes?
And another problem besides corners is that the surface area of the thing is wrong.
It's pretty close.
It's way off. Its volume might be correct, but its surface area surface area is going to be rather wrong, because you're going to have.
It's not going to be that far off, right? Because if you look at it, the reality might be this, and you're representing it like that. So it's a square root of 2 or something off, right?
Yeah, but it can add up.
Oh, sure. Yeah, absolutely. Yes, yeah.
But in general, what happens to the uncertainties, right? So originally you have these pixels and they have certain variations, but now whenever you do any transformation and even converting it to cylindrical coordinates, there is a Jacobian with that, right? And that the errors start transforming, and so the probability landscape is changing in every step, right? So how is that handled?
So so the way it's handled, and this is something we're very actively pursuing right now, is the first phase of this was to give us, or to try to generate at least one example of how to build a model of each of these different components, all right? We now have multiple ways of doing that, and now we're in the phase of estimating the overall most likely model given data, right? But then that means propagating all of these things and estimating the overall likelihood, right? Standard modeling thing to do. Yep. So, but it's hard, and so we're in the middle of it right now. Okay. So yes, please.
Is there also something in the reconstruction when you're trying to build these multiple organelles and that keeps them overlapping spatially?
So in the reconstruction, you're fitting them, and so they, like depending on what the model is, you're fitting them, and you might be fitting them such that they are not allowed to overlap, or they're only allowed, to touch but not overlap. When you're synthesizing, you can impose that as a requirement, right? And we do, in fact, say when you're choosing to create an instance, the first one gets plopped down, but the second one, can't lay on top of the first one.
So you have some kind of a rule there that tells you which one gets.
There's rejection sampling, that says you can't lay on top of somebody else.
In order to get the appropriate sort of relationship to numbers of different organelles.
Yep, absolutely, exactly. So that, and yeah, because you can't have the membrane boundaries, intersecting. Well, okay. All right, now, the problem sometimes, with the models I just gave you, one of the problems is they may not be particularly accurate, whatever assumptions I made, but one of them, even if they are generally accurate, You have cells like, for example, some kinds of, like neutrophils, have nuclei that look like Z's or S's or Y's. I mean, they're really funky shaped nuclei. And the model that I told you won't work for those, right? It will try to fit it, you know, to some, like the convex hull of that, which is not right. So we're going to introduce a non-parametric way of doing the modeling, and that is to just consider the distance, the difference between all of the instances of whatever it is that we're trying to model, and build a model just out of that. Okay? And the idea is that we take, here are, for example, one nucleus and another nucleus, just hypothetical, the 2D doesn't matter. It works perfectly, 2D, 3D, 4D, whatever you want. And we're going to ask, how different are these two nuclei? Now, can you have an idea in your mind of how you can measure how different these are? Basically, there are two kind of approaches. One, you could say, well, how many pixels are different? Or it turns out a better mathematically defined approach is to find a function that maps this guy into that guy, okay? With the minimum amount of change. It's a morphing function. It's like turning, you know, Schwarzenegger into the Terminator, right? It's just, I hope that was still, most of you know who the Terminator is. Some of you maybe not. Okay.
And then turning them into the Governator.
Right. Yeah, exactly. So we you solve a system of differential equations to morph this guy into that guy, and you take the integral of that. Okay? Does everybody have an idea what that of that makes sense? Right? And that might be different. This is the way you do this for shape. There are ways to do this for other things, but this is what you would do for shape. So now what we can do is do that distance for all the nuclei to each other. So far, so good, right? And the question is that what we want to try to now build is a model from it. And that model we can think of as a map where we put the nuclei that have similar shapes near each other and the nuclei that have different big distances away from each other. And that task is exactly like if we had the distances of all the cities in the US, but we didn't have a map of the US, we have to generate a map of the US. Right? And so we take our distances, and then we say, all right, here are the closest two, right? Put those two down, and then we add in the next one, and we just keep on doing that until we lay the positions out. We can just triangulate everything until we build up the map. That's assuming there's no error, right? And we can also do this problem with error. We can find the maximum likelihood solution. And then we build a map of the US, right? Or at least of those cities. So this is what we want to do for the shapes of things like nuclei and cells. Now, I mentioned that you can do this for 2D or 3D. It turns out you can do this mapping, this morphing measure, it's called a diffeomorphic mapping, for multiple components at a time. You can do it for nucleus and cell shape, which is how we're going to do it. And so what we're going to take is examples of cells with their nucleus and their cell shape. And then we're going to measure the distances between all the pairs and build up a distance matrix. I'm just showing part of it here. And then we use this distance to map approach, right, to generate a map of where of the various things. Now, here's the key point about this. And I'll show, and this is an example, a higher resolution example of that. This actually is a generative model for the following reason. If I can think of this as the space of all possible cells, and a cell that lives here, I can generate his shape by interpolating between his closest neighbors. All right? Do you see how that would work? Right? So I have, in this case, I'm talking about a two-dimensional map. It can be 3, 4, 5. It can be any number of dimensional, this shape space. I'm just projecting it in 2. And I can form the simplexes that contain any point and then project from one to the axis of the other and then synthesize an image to correspond to it and so on until I can generate. Does everybody, I can explain more about that. Yeah, you had a question.
How many components do you usually need to capture most of the
So the general problem is if I have, n shapes and I have n distances, I might need an n-1 dimensional mapping to capture all of them with perfect accuracy. So what's going to happen is I'm going to make some choice. In the field, typically, people do some kind of reconstruction error versus number of dimensions and find the knee. There isn't much better than that,
There are so many cells in the slide. Are they different single cells or same cells of?
No, they're different single cells. They're different single. So just randomly sampled from a field, different cells. Okay. Yes.
Could you give us some examples of the measures that actually measure the shape?
So the only measure is that morphing distance. It's to think of it as making a little vector. It's the deformation gradient, right? A little vector for each point in the first cell to where that point should be in the second cell, and adding all of those up, right? That's a simple way to think of it, right? So if you need a lot of vectors, you got to move a lot of stuff around, then that sum is big. And all it is just distance. That's it. One number for each cell, for each pair of cells. Yeah.
Then what
Is that
the transform function between one shape to the other?
okay? It's that. So from one cell, I have one cell and another cell, like the nuclei. I ask, which pixels do I have to move? So the pixels in the middle, they don't have to move, right? Because the middle of the nucleus stays the same. But the ones on the edges are going to have to move in order to find the right place on the other guy. Yeah?
But isn't that an ill-posed problem for non-convex shapes?
Well, it's not ill. There is always such a
Yeah, but it might not be unique, and therefore the distance might not be unique.
Absolutely correct. Absolutely correct. Yes. Yes. And if we can spend like 100 times the time of this lecture talking about all the things that are not perfect. What we're trying to do is to get to a point where we have, a workable solution, right? So we're going to have to test a lot of these assumptions, like we talked about, estimating model likelihoods and so on. And at any moment in time, which is the whole task of science, we have to pick the best model we have right now, right? And not worry about whether or not, there might be a better model. Yes, there might be a better model, but, we have to do something today, right? That's the fundamental idea of this. And this is this is a lot like, right now where we are, it's a lot like where we were 25, 30 years ago in molecular dynamics and like protein folding and things like that, where people started out by saying, well, assume it's kind of like, Lego connects, right? All the bonds are rigid, they're all the same length, and then you kind of work your way up, to learning more and more realistic, energy functions and so on, right? We're in that same space. We're back 25 years in dynamics. All right. So do you have a sense then of the answer to that question, of that you're morphing the two things and you're assuming that that the way in which you do that, like if they're very similar, you just push a little on this guy and that's it. That's the only difference between them. Therefore, you have a small distance. If they're really different, you're going to have a lot of ways in which you can move stuff around. So you're going to have a lot of solutions. There's going to be one that's mathematically found, depending on what algorithm you use. Yeah, but it doesn't have to be unique. All right. So now we can ask a good sort of thought experiment. This X marks the position of that cell. Now it's actually a three-dimensional cell, but I can only show 2 dimensions. Imagine what is it that cell will look like if I looked at that cell, that individual cell, a second later or a femtosecond later or whatever time scale you want, right? Is it going to look like one of these cells up here? No. It's going to look like one of its neighbors in the shape space. So I can do a random walk in the shape space and generate shapes that would to simulate the changes in shape of that particular cell over time. Now, I don't know that it's actually doing a random walk. It may be doing a directed walk, but it's moving over time in the shape space, and so I can actually start to simulate from asynchronous cells, from different cells. If I get enough of them, I can build up a map of the shape space such that I can say the probability that a particular cell is going to go in a particular direction. So this is random, which is almost certainly not what happens other than maybe in short time frames. But we can also think about measuring cells that are from movies where we know the shape of the same cell over time. Okay? In which case, we can take our shape space. I took the cells out. Now it's just based on the density of the cells at a particular time. And we can put in arrows that say, if a cell is at this position in the shape space, he's likely to go this way or that way in the shape space. Right? Yes?
Interesting that, like, let's say that you don't have that much data. Like, let's say you have 5 movies, and it's probably not enough to do the machine learning you talked about.
Yep.
Could you instead use each frame from the movie? So
Yes, and you would build a big shape space of all the frames, and you would have trajectories in the shape space that link each of the cells of one movie.
But could you use just those 5 movies to do what you talked about with the parametric models, or would it be not like sampled enough?
Well, if I had, for 5 movies of shape, I would do a non-parametric model probably. But then what you're going to have is you're going to have 5 trajectories in that shape space, right? Where the cell, for example, might have started out, here, and it's going to go clockwise or something, right? If all 5 of them show the same direction of movement, which is, sort of what's happening here, right? The arrows here, the arrows are pointing towards here, right? Then maybe I can, even from 5's stretching it, I could try to learn a model of what direction you should go depending on where you are, okay? Now, the point of this, I don't want to emphasize this too much, but it allows us to start to introduce dynamics into these models by learning spatiotemporal models, not just spatial models. Okay. All right, now I'm just going to give you some more examples. We actually covered a lot of the concepts that I wanted to get across, right? And the rest now becomes, some details as to specific models. But I want to give you a couple more examples of how this can be used. And this is a collaboration with Christoph Wolfing to study the spatiotemporal changes in signaling protein distributions when a T cell binds to an antigen-presenting cell. Okay, so when a T cell binds to an antigen-presenting cell, they form a T cell synapse that recognizes the antigen that the T cell is specific for. Okay? So the kinds of data that's generated is mixtures of T cells and antigen-presenting cells, where the antigen-presenting cell has its antigen stuck on the surface. And the T cell is labeled for one protein with a GFP tag. Right? So you have a particular signaling molecule, wave 2, cofilin, whatever it is, labeled in a particular cell line of a T cell clone. And then you mix them together and you take movies. This is a tiny piece of a field of cells where the T cell binds to the antigen-presenting cell, and you can see it start to happen there, and you get this interface. And then the task is to try to build a model of what's happening, of who's doing what, where the proteins are going spatially. Now, what you can see is the movies are fairly noisy, T cells are fairly small. And so if you were to try to build an understanding of how all of the different proteins were changing, it's hard to do that by just looking at these movies. So what we're going to do, I would take these fields, find individual cells, segment them, morph them to a half ellipsoid, because the cells kind of look like a half ellipsoid. So we're going to choose that as the normalized frame of reference for each cell. And then morph them, and then we can build up, by averaging, a model of 100 cells in that idealized shape. Does that make sense to everybody? All right? So here would be here's what we get, right? So these are slices looking from the side of the cell or looking down on the cell at before the synapse forms and at the time of synapse formation. For this particular protein, it's kind of just hanging out in the cytoplasm. And then when the synapse forms, it jumps up to the synapse. Okay? So that's for one time point. Here's another time point. So do you understand how we built these? We morphed them all to this thing that looks like an Eppendorf tube. And then we averaged them all together in that frame of reference, right? In that common frame. What that means is each pixel in that common frame of reference, at each time, we can string all of those numbers together. So we have a density or an intensity of that protein, a concentration of that protein at each voxel, at each time point. And that's a very big vector of concentrations. Okay? We can then cluster each protein and find proteins that have similar spatiotemporal patterns. All right? And what ends up happening is that this was done with, I think it was 12 proteins initially. And when we cluster them, we find 3 clusters. The vast majority of the proteins are in patterns that look like this. But there are also 2 other patterns and I'll show you what those look like. So what we're going to do now, so we've clustered all those proteins, and we're going to just show 3 patterns. And we're going to show those 3 patterns as a movie of concentration versus time. Okay? And the colors, each protein has a color, red, green, blue. And where it's purple, that means the 2 proteins are the same. When it's white, all 3 proteins are there. And that is a movie of the changes in concentration of these signaling proteins during the process. Now, my collaborator loves this movie. It makes sense to him. I think it's too confusing. So the way I like to look at it is to threshold that to show you the regions where most of the protein is, where the protein has a high concentration. So here's an example, right? It's about to switch over to, oh, I got to rewind it. Right? So it's uniform, and then it starts to move up to the interface, hangs out on the interface for a while, and then starts to diffuse back down. Okay? So that's the simple pattern of that first major cluster. Well, we can do that in multicolors. And so this gives you a little bit better feel of what's happening. See there, the blue guy is never jumping up as much as the white guy, as the other 2 are. Right? It's much delayed in terms of the time that it goes there. All right? So this is just showing the clustering. And we can actually show how the distributions change when we block signaling, for example. All right, I may skip over this. This is an interesting one, but it's maybe not as relevant. This is actually building a synthetic model of the differentiation of a cell from a rounded cell into a neuronal cell, and the details of that are maybe not so important, but the idea is that we can build these spatiotemporal models that capture things like the differentiation process for a cell. Then sort of the next frontier is going to be to try to be able to do cell simulations in these dynamic varying cell shapes. Okay, so bottom line is we now have the early versions of systems that can build models of components of cells and learn the relationships. I haven't talked about that. Learn the relationships between them. So, for example, we can build models of nuclei and ER and in a plant cell, chloroplast, for example, and then we can ask, what's the distribution of ER relative to chloroplasts? Or what's the distribution of ER relative to Golgi? Or mitochondria relative to microtubules, and so on? And build up higher and higher conditional models. And we already have started doing some of that. Right? And that's the task for the next 20 years, is trying to build up all of these components of how they all fit together. So what does that do for us right now? what you can do, and I guess you're going to hear more about this in a bit, is we can take models like this, and one of the things that's the characteristic is we can at least make the guess or the assumption that a model that we've learned in 3T3 cells for lysosomes and mitochondria may be equally applicable to, HeLa cells. Right? So we can actually potentially mix and match models that we've learned either from completely separate cell populations or even different cell types, and at least potentially combine them, and then we can test whether, for example, the combinations actually correspond to what we would see with particular images. But it's a much simpler thing, or much less work to test one of these assumed models than it is to build a model. So we can. Does everybody understand what I mean by combining that? So I can. So it's like a Rector set or whatever. I can say, give me a nuclear model from this, and then give me a cell-shaped model, and then I'll take model from cell type A of this protein and cell type B from this, and potentially I can combine those together. Right? Furthermore, we're building up libraries of these models in different cell types, and it might be that for a particular simulation, you might be interested in one of the cell types you already have a model for. Okay? And furthermore, obviously, if you're doing experiments yourself, you can build your own models. Right? You can take images of your particular cell type for your particular organelles or proteins or whatever and build your own models. All right? So I think I'll stop there and see if there are any more questions, especially more conceptual questions of, how can we use this, how can we apply this, and the kinds of things that you're particularly interested in. All right? Thanks. Yeah?
How good is it, or would it be even at the stage where you would have a lot of constraints between the cells? Let's say you want to grow a little bit of brain, then all these neurons, grow, and they have these boundaries, and they, it becomes very much more complicated than these isolated, nice little problems.
Yep. So this is another example of the mix and match thing I was talking about. There are people who work, and there are reasonable efforts at models just of the shape of cells as they form tissues. Okay? So that just looking at that, you take a liver or whatever, and you build up a model that would allow you to synthesize a liver, for example. But that liver, those cells, don't have anything in them. They're just like boundaries of cells. Well, this is a way that I can put a nucleus and secretory vesicles and whatever it is that I need in that tissue into those. Once I have those shapes, I can use that to instantiate all the different organelles. Right? So that's an example of combining these things. By the way, if it wasn't clear, we can take these geometries and draw an instance from them and then do biochemical simulations in that. So for example, a lot of the time, people will build up a complicated biochemical model, and it could be even compartmental. It might have things that are going into different parts with a cell, and there's rates of reactions and so on. But then run that model in, a sphere inside of a sphere. Right? Well, we can pick one of the cell types out of Cell Organizer, and then you can run that model in a realistic geometry. That's the fundamental idea. Yeah? I think you had a question.
Yeah, I did. I really like the mix and match idea. I think it's like a great step between, like you said, a circle and the mirror images. I'm assuming the libraries you talked about are available now?
Yes, they are. So when you download Cell Organizer right now, you get all the models that we have trained already. And of course, you can train your own. Right? What we're setting up is a web page where you can go and find models, right? That maybe somebody else trained. So you train one, and then then you put your model of, whatever cell type, and so on. And then what we can also do is that we have some tools, some initial versions of tools where if you want to build a compartmental model of X, Y, and Z, you can ask Cell Organizer, do you happen to have models of X, Y, and Z? and then it will find them and pick them and then synthesize, right? Does that make sense? But it's very early on, and we need to do more and more of this. And I'm hoping that some of you guys will actually build some models that you can then deposit and that more and more people can use. Yeah. Yes?
A few of us here are interested not in the cells, but the spaces between them. In other words, the extracellular space, the
Yep.
matrix, and so on. And that is proving to have quite a complex geometry. Do you think there's a future for this approach in reconstructing rather large areas of extracellular space?
Yep. So yes is, of course, I think the answer. And then the question is how, right? If you have models of the cell shapes, then you can just do exactly the same things we've talked about and build a model of the extracellular matrix conditional on the cell shapes, right? So that whatever geometries you have in between cells, right, that's a coordinate system that you can learn a model from.
The problem particularly on the nervous system, and half the cells there are real cells, which have an incredibly complex geometry. So this would be rather a tough way to approach that problem.
Well, okay, but you can also consider each intracellular space as an entity of its own, right? So if it's bounded by cells, right, then that is an entity you build a model for. Now you have a model for whenever you have a space, you can put, and it can make it as dependent as you want. For example, it can be, dependent on the region of the brain. It can be dependent on how big the region is, and so on and so forth. I didn't really emphasize it there because there's a type of model I didn't talk about, which is a forward model. So for example, if you want to learn a model of the distribution of microtubules, you can't really do that by the methods that I talked to you about, right? So what instead we do is we build a model where you grow your own microtubules from the Microtubule Organizing Center, and you grow your own, and you learn the rules for growing them, right? So that's kind of a rule-based, it's not exactly rule-based, but it's kind of, you can think of it that way, as a generative model of growing microtubules. Well, that may be the right approach for extracellular matrix, that you don't just have a statistical, texture model or organelle model or whatever, but rather that you learn various ways that you could grow the extracellular matrix, and you choose the model based on your images, right? So you would choose that model which most resembles or the parameters for that model that most resemble your real images. Does that make sense? Yeah, back there.
You talked about the intensity analysis that you do. I don't know if I caught it. How do you convert those intensities into actual numbers of models?
Right. So that depends on the specifics of the system that you're working with, right? So in some cases, people have calibrated fluorescence to molecules. In other cases, you know the total number of molecules, and then you just sum the intensity distribution and normalize it, right? If you know that a cell has, 1 million molecules of X, and you know the fractional distribution, then you have the concentrations.
How do you do it for images where you if you have slices for cells? How do you account for overlap between the slices in terms of fractional intensity?
Yeah. So that's all part of the reconstruction phase, right? So remember we talked about there are two phases, which is I have one image or a stack of images, and I'm converting that into a generative representation of that. And then I'm building a statistical model. So that first part depends on what it is that my images are. And the short answer is that, again, we can make the assumption that that's going to wash out. If I normalize the total intensity, sum it to 1, that there's overlap, but it's going to get normalized out. I'm doing some double counting, but I'm doing the double counting uniformly. Do you follow me? if I have overlap between the slices such that I'm measuring intensity from the below side, depends on what kind of microscope you're working with, right? Then I'm counting sometimes intensity in this slice that was really in the other slice.
Yeah.
And I can deconvolve that, of course, if I know what the point spread function is. But I can also just essentially ignore it because I'm counting this, the fluorescence from this one and that one. I'm counting the fluorescence, so I'm double counting, but in the overall, it's going to wash out. Yeah? Approximately. The main point there, though, is whatever the source of images are, you should try to get the best representation from that image. So if you want to deconvolve, or if you're going to use super resolution microscopy or whatever, such that you're starting your process of modeling with the best example you can. But that may not, the best may not be that good, but you can still get a reasonable, especially because you're sampling over many, many, many cells, right? So, yeah, okay. Tom, did you have a question? Or?
Yeah, well, it was related to what Charles was saying.
Yeah.
For generating that extracellular space, especially in neural film. If we look at it in the inverse way, which is what are the shapes of the dendrites and axons and their relationships to all their and the dendrite spine and everything, the relationship between all those things together. They come together and they have these really complicated shapes that have to fit together like the crazy Chinese jigsaw puzzle on 3D.
Yep.
And then they're surrounded by the extracellular space that they all create together. I'm just having trouble imagining how.
Yeah, and I mean, you just have to, I think, that path on that path lies madness, right?
Yeah.
You just have to break it down,
Yeah.
right? There are people, and we are also building models of dendrite morphology and branching and so on, right? There are people who've been doing that. So you say, well, for right now, I'm going to assume that's a good model, right? And I'm going to use that model and throw those together, and then I'm going to generate spaces, and then in the meantime, I'm going to learn models of the spaces, and I'm going to try to see, and it's iterative, right? And like we talked about before, I can compare two different modeling approaches and say which one's better, at which one has a higher likelihood given data.
Yeah, and then that last bit, given data,
Yeah.
is also a huge sticking point because we don't have a lot of instances of ground truth.
Well,
of
but
what the thing really is, right? In the machine.
Right.
Truth in the machine.
Well,
So I don't think you're going to get it from individual images. You're going to get it from these very painstakingly made hands.
no, because that assumes that a person knows that reality, and a person usually doesn't. They think they do.
Yeah,
That's
but
what 20 years of this field has
There
shown.
any machine learning,
okay.
machine vision
All right, so
that can extract
Yep.
the cell membranes from
Okay.
neural film.
Okay, so
If we had
now
that,
there are
then-
two possibilities, all right? One of them is the task is impossible,
Yeah.
okay? The other is it's possible. Now, 20 years ago, I spent a good number of years convincing the cell biology community that it was possible to quantitate images and recognize these patterns on the descriptive side before we start on the generative side. 90% of the people that I talked to said exactly what you said. Oh, we don't have the ground truth. You need to go to graduate school to learn to recognize the difference between an endosome and a lysosome or whatever. It's just not true, okay? Once we have data, once we use it and choose an appropriate learning strategy, we can learn models that perform better than people do. It's just a question of doing it, right?
Yeah.
And we often thereby discover that the ground truth, if you then do an experiment where you have a way of getting the ground truth independently, that ground truth as judged by a person is not the same as the ground truth that you learn experimentally, for example. So, if there's one thing that I would hope that you guys would take out of this, It's that process of hand tracing things, while it may be the only thing we have available now, we've got to do better than that.
Well, that's
Right.
for sure.
Yep. And
But,
in that
but
particular,
that
yes,
one thing is the impediment to
that's right.
getting to the
And
gender.
if you care about a particular, like in brain, if you care, or, if you care about a particularly complicated geometric system, then you're not going to be in as good a position as somebody that's working, on cultured cells or, or even a less complicated tissue, right? Because it's going to be easier to learn models for that tissue,
Sure.
right? But part of the point of the center, the ML Biocenter, is that when people have specific problems like this, you want to learn a model of extracellular matrix, and if you have data, images of various proteins that are part of it, then the center is looking for projects like that where we would develop a new module that would be specific for learning extracellular matrix, or would be specific for learning, whatever you particularly care about. Does that make sense?
Same thing.
Yeah.
So when you say that the machine is much better than the humans in doing that, then what, and there is the ground truth doesn't come from the humans, how do you know that,
Yep. So all right. I'll tell you a story. I'll tell you a story. Back in the days when we were training systems to recognize all of the subcellular patterns. Okay. What we did is we said, I'm not going to assume I know. I'm going to just take images of particular proteins, so I know what protein I labeled. That's the real ground truth. All right. And we took proteins that were thought to be markers of the Golgi and lysosomes and endosomes and mitochondria and so on and so forth, then took images of them, and assuming we didn't add the wrong antibody or the wrong marker, we knew the ground truth. We knew all of these images are mitochondria, are of this protein. Right? Then we trained a classifier with that ground truth, and it nowadays it can do 99% accuracy on that task. People do about mid-80s on that task. So then people said, well, how do you know well, you know the ground truth of those proteins. And it turned out that two of those proteins were both Golgi proteins, which people couldn't tell apart. So people said, these are both Golgi proteins, but the system could tell them apart with 99% accuracy. So it was learning that the ground truth was more complicated than people thought, because people would have put them into. Right? And the funny thing is that there was another study done on this task that generated images, but they had people assign the ground truth, even though they knew what protein it was that they had labeled, because again of this notion that. But I think the key is designing the experiment in such a way that the experiment contains the ground truth. Yeah.
But then you need some marker, right? You need a molecule which is in the domain you're looking at. So in brain extracellular space, you have regions where there is some matrix, but there are some regions where there's probably none matrix whatsoever. So then if you go with your marker, you're going to pick just parts of the brain extracellular space, but not everything. So what is the marker for extracellular space? It's water, it's sodium, it's chloride.
Yep.
So how do we go about that?
Yeah. So I want to just contrast the pattern recognition, which we're talking of as an example of where you have to have ground truth in order to train a system, from the generative model building, which in a sense is learning the ground truth for you, right? And so in that case, if you're trying to put together cells in a tissue, whether it's brain or anything else, you're learning the conditional model of every molecule on every molecule, including, water, right? I mean, like water, everything's, like you'd assume there's water everywhere, and then you're learning all the other parts. And so you don't need a marker if there's a region whose marker is the absence of markers, if you follow me, right?
Well, of course, one approach to this problem considering is to put in a fluorescent molecule that just remains in the extracellular space,
That's fine.
polarizes, and then dissolve away everything else.
So, yeah, so
Yeah.
An
But-
idea I had for doing this was to use something like biocytin and fill the extracellular space with biocytin and then activate it and it turns black.
Right. So,
And so the only way then to look inside your tissue then is with
okay, so we're distinguishing two different things here. One of them is a model of the extracellular volumes, just the volumes. And I think it would be wrong to try to learn that model directly, because that model is the product of the cells crossing, right? The spaces are the places where the cells aren't, right? And so if you're just caring about the geometries of those intracellular spaces, that I would generate by generating cell models and letting them cross. But if you care about the organization of things within that space, like, you know, how far out from the cell some particular extracellular matrix one protein is, and then another one is sitting on top of that one, and then another one is sitting on top of that one, those models you have to learn in those spaces. But there you would do that by marking each of those extracellular matrix proteins. Does that make sense, or is that?
It's a combination. I think it was a Chinese philosopher who said that a vessel is defined by the space inside it, or the void inside it.
Yes.
I think we look at it more from that point of view, from a very extracellular space centric perspective. But of course, with the the technical problem is preserving the extracellular space. When you can fix tissue for EM and do it, but you usually reduce the extracellular space from 20% volume fraction to 5% when you do that.
Mm-hmm.
And of course, not uniformly, as has been shown by the work by Tom and Kinney and other people. And so you don't know how it can differentially shrink. So you really want to preserve the non-uniformity. For the modeling purposes, this happens to introduce very significant effects on diffusion.
Right.
And so these are the sort of questions we wrestle with. And the matrix is a component of that. We're very interested in the matrix in and of itself within this space, as well as the space itself. They both contribute to the physiology.
Yeah, so they're dual problems, right? You can learn it either direction. The only reason I was saying that is because there has been much more work on models of the cell geometry than there has been on the, right? So, you know, that's probably better to start from. Any other, yeah, back here, and then over here.
I have a question. Isn't it easier to use Bologna analysis to keep track of the nucleus and then draw the line, the Bologna, and find the shape of the cell roughly, and then find the
Yep.
softer shape?
Yes. So that is another parametric model, right? You would be creating a model that would consist of the vertices of that Voronoi. And for some cell types, that might be a better approach. And one could add a module that does that. So if you were working with a cell type that had that, right, the way Cell Organizer would handle that is that you would set a parameter that says, for these cells, learn model X for cell shape, right? And then you could do that. And then, as we were talking about earlier, you could compare the quality of those models to models that were diffeomorphic or models that were, you know, ratiometric or whatever. Yeah, absolutely. Yes, over there.
When you use your models in cell simulation, do you usually run, I mean, maybe this is more of a cell technical question, do you usually run like multiple seeds from the same model, or do you run individual simulations from different models? And I guess to
Yep.
what extent do you observe like significant?
Devin, are you going to be talking later about the stuff that you did, or just do? I mean, what's happening after this?
I'm going to give a brief overview of sort of like how if you actually have data, how to get it into Cell Organizer more technically, like what sort of pre-processing steps there are. I hope that'll be really brief since you covered most of the technical aspects. And then I was going to talk a little bit about some of the work I did on both dynamics and neurons, because a lot of people here are interested.
Okay. So the short answer is that these are random, they're random simulations, or they're, right? You have to sample different geometries, and then you can look at the effect of your simulation, in your simulation, of geometry. That's one of the key things that, you can do for a given biochemical model. You can simulate the same biochemistry in cells of different shapes from that shape space and ask, do I see differences in the performance of the model, that's, the rate at which some event happens, depending on the geometry. But in order to do that, you have to do multiple seeds within that geometry so that you have a robust estimate of the behavior of that geometry. So both, yes, which does mean computing.
Yeah. And because all the simulations are independent of each other, they're individual simulations. The different cells are different cells, and the different seeds are separate runs. You can do the whole thing in parallel. On a compute cluster. If you have to compute, right? But that's what the big computing centers are about, right?
And
you have those resources, and you use those resources.
There's also a technique where you can try to do more simulations in a region of the space where you think things are more different.
Rory talked about it.
Oh, Rory talked. Okay, good. Okay.
And that would be the weighted ensemble.
Right. Okay. Well, I've probably taken up too much of your time. Thank you very much.
Maybe we'll take a short